A sua apresentação é regida pela portaria 45 de 2025, né, que resolve designar os professores Humberto José Lira Queiroz, sou eu, professor Cleomar da Silva Moreira, do IFPB, que está ausente, não pôde participar, mas mandou parecer, professor Carlos Alberto de Souza, da UFPB, que está presente, juntamente com os professores Antônio Marcos Nogueira Lima, que é seu orientador, e professor Aprígio de Melo, Arthur Aprígio de Melo, né, seu outro orientador, também da UFCG, para junto constituir a comissão avaliadora do seu trabalho, né, de Rodrigo Toledo de Araújo, o trabalho intitulado Modeling Sensorgrams Issued by a Surface Plasmon Resonance Biosensor, cuja apresentação se dará a partir de agora, então. Rodrigo, desculpe interrompê-lo e agora sim pode continuar a sua apresentação. Ou iniciar novamente.
Tranquilo, professor. Vou recomeçar aqui. Good afternoon, everyone. My name is Rodrigo Toledo de Araújo, and today I will be presenting my master thesis dissertation. The title is Modeling Surface Plasmon Resonance Biosensor Sensorgrams. I would like to say thanks to my two advisors, Antonio Marcos and Arthur Aprígio, and to the examining board. This work was made within the Department of Electrical Engineering in the University of Campina Grande with collaboration with Virtus Centro, Virtus Competence Center. So the summary will be a guideline for our presentation. I will start with an introduction. I will bring a contextualization, innovation, the validation and objectives of my work. Then I will talk about the SPR absorption models that we developed. I will talk about the heterogeneous, the linear regression, the recursive least squares, the physics-informed neural network models, and some experimental challenges that I had. And in all of these methods, I will talk about the methodology and materials and results. And then I will talk about the final considerations, some discussions about my work. The introduction will start with a contextualization. The main objective of my work is of this project is to develop, implement, and validate a suite of computational frameworks for the analysis and simulation of SPR biosensor data. To understand this main objective, we first need to understand what is a biosensor and a sensorgram. A biosensor, like I'm showing in figure 1, is this analytical device comprising a bioreceptor and a transducer. When the analyte interacts with the bioreceptor, some physical chemical changes happens, and these physical chemical changes are then transformed by the transducer into a signal, a measurable signal. This signal is then processed by the signal processing unit. It can be a computer, and in our case, this signal is the sensorgram. So this SPR biosensor data is the sensorgram in our case. So we were trying to model this sensorgram, and in this next slide, I will talk about the SPR transducer a little bit more deeper, because in this slide I just said that the transducer made the change from the physical chemical changes to the signal, but here I will talk how the SPR do this, the SPR biosensor. Here I got a angular interrogation mode SPR biosensor. We got 4 layers. The first layer is an optical substrate. The second layer is a metal, typically gold. The third layer is a recognition membrane. The fourth layer is the analyte that we are trying to measure some characteristic. And in this operation, we got a light source, and this light source will emit a light beam into the metal, and when the light beam strikes the metal, the electrons in the metal surface will be excited, and this will generate a surface plasmon wave. And this transference of energy between the the light and the free electrons in the metal surface will then be seen as a dark region in the reflected light. And we can detect this signal by using, for example, some photo detectors or a camera. And this signal is then passed to a signal processing unit. This will be like a SPR image, like I'm showing in this figure. And this SPR image is indeed a matrix of pixels, and if we analyze each line of this matrix, we can then build these reflectance curves. This is the reflectance over, for example, the angular displacement here, because we are dealing with the AIM biosensor. And when we analyze the minimum value of this reflectance curve through the time, we got the sensorgram. So the sensorgram is this SPR biosensor response through the time. And talking a little bit about the sensorgrams, they are this response of the biosensor to these analytes and these substances that we are trying to measure. And this interaction is indeed a adsorption process when the analyte, for example, binds with the metal surface. So the adsorption process is a really important part of my work, but it is also really complex to be modeled. This happens because each model can absorb into a surface in a different manner. So it is really hard to fit this process into only one model. We have several models, for example, the Langmuir model, the random sequential adsorption model, and even models with more than one state for the molecule. But when we talk about this process of adsorption in an SPR assay, this is typically used to form a recognition membrane. So this recognition membrane is like the when we try to make our sensor really focus to one specific target. So it's like a functionalization of our surface, where we are trying to make the biosensor really specific to certain molecule and not to any kind of molecule. And then the specific objectives of my work are to develop and formalize a spatially resolved computational model to simulate the heterogeneous biomolecular interactions on a sensor surface. This will then enable the generation of kinetic maps, SPR imaging, and multi-site sensorgrams analysis. Implement and validate a recursive least squares algorithm for the real-time estimation of kinetic constants from SPR sensorgrams under the Langmuir assumption. Also, design and train a novel physics-informed neural network architecture using long short-term memory cells to provide a robust AI-driving solution to the inverse problem of kinetic parameter estimation and prediction problems from noise data. And also to validate these models and other linear estimation methods using data sets from scientific literature and some experiments that we made it in our laboratory. Now I'll start the second part of my presentation about the models. The first model that I will talk is the heterogeneous model and what can be accomplished with this model. When we integrate the SPR transducer with an spatially resolved surface like the one I'm showing, the animation one, we can then even find kinetic constants for these sites and also generate sensorgrams and SPR images when we integrate these two particular models. And then we can also build an end-to-end simulation of an SPR imaging experiment. So we can see this in the animation 1. We got, for example, a flow cell here. We got the fluid that is going from the left to the right. We got that the surface is being the phenomenon of the adsorption is occurring in the sensor surface, and then we can then build the line sensorgrams, for example, for each line of the surface, and we can also build the SPR reflectance curves and the SPR image. So this is a forward process and also an inverse process, because we can go from the sensorgrams to the image and from the image to the sensorgrams. The assumptions of this model are the following. We got a discretized surface like in the figure 7. And in this discretized surface, we got different sites. Each one of these sites, we are considering a local Langmuir kinetics. And the sites can be different, and this way we will call of a heterogeneous parameter field, meaning that each site can have a different set of kinetic parameters. The fluid flow is modeled by the advection diffusion transport, and we also are considering Gaussian measurement noise into the data. So as we can see in animation 2, we got the sensor surface coverage, a top view, and from each one of these lines, for example, we can build these sensorgrams for each one of the sites in one line. And when we sum each one of these contributions individually, we can then create a observable we can create an observable line sensorgram for each one of these lines over here. So we got this procedure in our model. And it is important also to quantify the identifiability of the parameters in this model, because it's important to, from measurements, from experiments, to correctly identify the parameters, the kinetic parameters from the model. So for example, if we are considering a line heterogeneous parameter field, like I've shown in the previous animation, we can then consider the following line observed sensorgram, and then the estimation problem becomes a nonlinear least squares algorithm, for example, we can use, and the estimation problem becomes to minimize the error between the model prediction and the data in order to find the best set of parameters to minimize this residual. So we define the parameter identifiability for a set of parameters P. This set of parameters is, we say that they are structurally identifiable for M experiments if these two conditions are satisfied. The first condition states that the number of points that we have must be greater or equal to the number of parameters, and that the Jacobian of the parameters, this is the partial derivative of the response in the partial derivative of the response in respect with the parameters. The rank of this Jacobian, this is a matrix, must be equal to the number of parameters. And here in the animation we see that this has been done in using some data. And then here I'll talk about the data generation, the data that I generated to validate this model. We use a neutral molecule. We got some parameters for this molecule. We got parameters for the light source and parameters for the biosensor, and also parameters for the fluid flow cell. And using these parameters, we also built this, in figure 8, we can see the different types of surfaces that we design. We got a fully homogeneous model, a fully heterogeneous model, and also the line heterogeneous model. So we tested with all of these surfaces to see the results, and we generated these multipose sensorgram to identify the parameters and validate the model. And a question that someone may ask is this data sufficient to the problem of identifiability of the parameters? Then we made the singular value decomposition analysis, where we analyze the singular values and the parameter contributions to the singular vectors. We can see that the singular values, they are related to the magnitude in which the parameter influences the model output. So if the parameter has a big singular value, this means that the parameter is important to the output. So we got a 3 degrees of importance in terms of the parameters. And we also analyze the singular vector for all these parameters, and we notice that these singular vectors, they are linearly independent. This states that that using this data, it is sufficient to really identify the parameters. And here we got some results for the full heterogeneous model. We are in the figure 10 comparing the observed sensorgrams from a fully homogeneous surface and a heterogeneous surface with 5% of random parameter variation. And we can see here that when we got a surface that has a larger number of sites and the parameter variation is low, in the end, the result will be quite similar to a homogeneous surface, because the sensorgrams will be kind of similar between the sites. So we got this result with just a little bit of difference between the homogeneous and the heterogeneous model. And we also analyze the decomposition of the observed signal with a non-zero initial condition. We can divide the response, there is this black curve, into two terms, a decay term and a kernel term. So this decay term is related to the already bound molecules. They will exponentially decrease with the time, and the kernel term is related to the new molecules that are arriving and the molecules that are also dissociating with the time. And here we analyze two scenarios, one scenario with noise and the other scenario with no noise, to see if the model can really identify the parameters, the true parameters. And as we expected for the scenario with no noise, we got that the model recovered the true parameters with a really small error. We can say that the model recovered the true parameters in this case. And for scenario B with noise, the algorithm didn't recover the true exact parameters, but recovered an estimated a set of parameters that was inside of the interval of confidence, 95% of confidence. So here we can say that the algorithm it was also successful, but because we have noise, that the parameters was not the recovered parameters were not the exact ones, and we can see that the residuals are mainly Gaussian noise as well. And also we tested with the line heterogeneous model. This model has less parameters than the fully heterogeneous model. In this model, only the lines are different. Only the lines has different kinetic parameters. And in this kind of approach, we had a better result. So we got that the maximum error between the estimates was about 0.8%. So this was a really low error compared to the other solution. So even with the noise data, we got this estimation, and we also got the residuals being the being only noise, mainly noise, when we compare with the result. And here we in figure 15, we are comparing the sensorgram calculated directly from the physical model. So the sensorgram made it by the forward approach using for example, the model from Langmuir, and a reconstructed sensorgram from an image that is also a generated image, an 8-bit image. So we are trying to see if the forward the forward method match the inverse method. So we can see that they kind of match the blue and the red dotted curve, but what happens here is because we are passing from an 8-bit image, we need to use only 256 possible values to the reflectance. So we are not dealing with a continuous array. So we got this kind of discretized look into the data, but we can see that we can do this inverse approach as well. And here I will start talking about the linear regression models and what we can accomplish with these models. We know that from data, we can, knowing the model, for example, the Langmuir model, we can fit the best possible model, meaning that we are going to find the best set of kinetic parameters to fit the data. So what we can accomplish, we can derive kinetic constants from data, and then produce sensorgrams and other representations. So the data acquisition for these models was made by analyzing some literature, for example, papers from Laborde and Trishap, and then we collected these papers. We analyzed the data. We then digitized the data using some software and got the data. Here we got some results for the biotin. We made it the linear regression, and then we found this set of kinetic parameters. We then plot the isotherm plot with the regression, and using these parameters, we could also plot some sensorgrams for the biotin with this concentration. The same was done for the NaCl molecule. We made it the linear regression, then we plot the isotherm, and we collected the kinetic constants, and then we made it the sensorgram as well. Now I'll talk about the recursive least squares model and what can be accomplished with this model. In this model, we can apply this technique in a real-time experiment, for example, and find kinetic constants and then predict the sensorgrams responses. So we just need to discretize for example, the Langmuir adsorption model and apply the RLS technique, and through the iterations, the kinetic constants are going to be estimated. And the data acquisition procedure was an experimental one. In this case, we got some stock solutions, and then we diluted these solutions. We made it some solutions of NaOH for different concentrations, PBS and also HCl. And then we analyzed these concentrations using an SPR biosensor that we have in the laboratory. And here we got some results for the PBS. Here we got in the figure 25A, we got the prediction and the sensorgram prediction for the last estimated kinetic parameters, set of parameters. And here we got the evolution of the parameters through the time. We can see that given a certain time step, the set of parameters converge to a certain value. And we also can see that the least squares error is low in the order of 10 to the minus 7. And also for the NaOH and HCl, we got the results in figure 26, and we got some least squares error in the order of 10 to the minus 5 into the order of 10 to the minus 7. We also estimated the set of kinetic parameters. Now I will talk about the physics-informed neural network model and what can be accomplished with this model. Differently from the traditional approaches of physics of neural networks, the physics-informed neural networks, they try to enforce in the training process some physics that we already know. So it is not only driven by data, we are also considering physics and some phenomena to model and to train the neural network. In this case, we can train this physics-informed neural network without knowing the constants and predict the sensorgram responses and also the kinetic constants. And then we can also, in the future, generate a sensorgram database, for example. About the data generation for these physics-informed neural network, we generated the data, so we deal with different sensorgrams, all of these sensorgrams with different initial conditions and concentrations. They all were made using the Langmuir adsorption kinetics. Then we added some noise into these measurements, and then we divided these different sensorgrams into a training data set, validation data set, and a test data set. And the architecture and loss function of the physics-informed neural network are shown in this figure 29. Basically, in the neural network architecture, we got two different main parts. We got a initial states neural network where we're gonna map the hidden states and cell state into the long short-term memory neuron, and this neuron will then predict the output. This is the sensorgram and new hidden states and cell states. So this neuron is not like a perceptron neuron. We got that this neuron is has these terms of memory. This is the hidden state and the cell state. So this is not a typical neural network structure, and it is suited to the problem of time series data, like the sensorgrams. And then after a epoch, for example, we got the the predicted sensorgrams and the estimated set of parameters. With these predictions, we can evaluate the loss function. Here we divided in two parts. The loss function got a only data part, data-driven part, and another part that is related to the Langmuir ODE equation. So this is the part that has the physics. So we evaluate this loss function, and if the loss function is not what we wanted to be in order to in terms of the value of the loss function, then we can update the weights and the parameters and make another epoch. So here I got the results from the training procedure. In figure 13, we got the loss from training and validation, the separate loss for only data and physics loss, and we see that they converge after these 40,000 epochs. And we can see also the progression of the kinetic constants, and they assume in the end an error of 4% and 8% when compared with the ground truth values. And then about the results and the performance of the physics-informed neural network to the data set, we can see in figure 31 we got the coverage and the derivative of the coverage. We got the predictions in black, and we can see that the residuals of this the residuals between the model prediction and the data are mainly Gaussian. And this is also confirmed by the Shapiro-Wilk test, where we can say that we rejected the null hypothesis. This means that the data, that the residual is mainly noise and Gaussian noise. Now I will talk about some experimental challenges that we had. The first one was the calibration. In the figure 32, we got the influence of the calibration at the sensor response in a biotin SPR assay. So we got the biotin, and we got PBS, and then in this first experiment, in this long experiment, we got biotin and PBS, and then we got the cleaning cycle, and then we have done this 3 times, but only with one calibration. And in the other row, we got the same cycle, but we have done 3 individually calibrated calibrations. So we wanted to see if by doing this individually calibrations, we had different adsorptions. So in figure 33, we are showing the correlation matrix of the experiments and the principal component analysis of the experiments. And here we can see in the correlation matrix that when we do only one calibration, and it is not a really good calibration, we can see here that the correlation of these experiments, especially the experiment of the part 1, we got a really low value when compared with the other experiments, for example. And this is also confirmed by the principal component analysis. when we analyze 4 features from the sensorgrams, we got the maximum signal, the mean signal, the standard deviation, and the area under the curve. When we do the PCA algorithm, we got 2 principal components from these 4 features, and we can see that the experiments that we have done, the calibrations individually, they group. So this shows to us that these experiments are more related to each other. On the other hand, the experiments with one calibration, they diverge from this group. We also face some experimental challenges with the materials quality. For example, in figure 35, we can see that a biosensor chip can have the gold layer degraded by usage. So this phenomena we can see here in figure 34, the influence of this degradation. Also for a assay of biotin, so we got some really different results, even with this even making calibrations in each experiment. We got different responses and different absorptions because the degradation of the gold layer. So we also can see this numerically by the correlation matrix and the principal analysis in this graph. They are really far from each other, the experiments. And now I will talk about the final considerations and some discussions. So in this presentation and in this work, we developed some SPR models. We implemented these models and we validated these models with experimental or generated synthetic data. The heterogeneous model provides a spatially resolved simulation of the sensor surface, and this can enable the SPR imaging and multi-site sensorgram analysis. The linear regression models can estimate the kinetic constants from experimental data, for example, and the recursively squares model enables this real-time kinetic estimation from experimental data. The physics-informed neural network model proved to be robust in solving the estimation and predict the sensorgram for problems with noisy data. And we also seen that we got some experimental challenges that confirms that failing to recalibrate introduces large variations, while individual calibrations produces consistent data, and this can directly affect the response. This is the sensorgram, and the absorption model can really suffer from these issues. And for future work, we are trying to, and we will try to integrate more advanced optical models, such as those based in Fourier optics, along with detailed image sensor models like noise models for the image and the camera, for example, and also a simulator of an SPR imaging instrument to then generate synthetic data sets to enable the training of deep learning models for direct kinetic analysis. And now I conclude my...
Ok, Rodrigo, obrigado pela apresentação. Foi no tempo razoável. Vamos dar início à segunda fase. Pronto, né? Bom, Rodrigo, então a comissão avaliadora deliberou e decidiu pela aprovação do seu trabalho. O conceito atribuído ao seu trabalho foi de aprovado, certo?
Certo.
Parabéns pelo trabalho. E agora sim, você tem a palavra para apresentar as suas considerações.
Antes dele falar, eu só queria, quer dizer, fazer um comentário final sobre, primeiro, agradecer a colaboração de Arthur aí na parceria de orientação, né? E, bom, parabenizar também Rodrigo, né, por essa aprovação, né? É um trabalho extenso aí, quer dizer, bastante complexo, né? Então, parabéns, Rodrigo. Tá? E obrigado a todo mundo que assistiu a esse evento, o presidente da banca, o professor Carlos, o professor Cleomar, tá certo? E destacar um pouco essa iniciativa que já tem alguns mestrados que eu tenho forçado o pessoal a escrever o texto em inglês como estratégia de internacionalização. O Rodrigo aceitou essa responsabilidade. E aí dessa responsabilidade derivam algumas coisas que estão indefinidas, ou seja, não existe um padrão da Copel, você sugeriu que a gente procurasse o padrão Copel, mas não existe um padrão Copel para texto escrito em inglês, a gente está tentando construí-lo, né? Eu fiz um template do LaTeX para ser utilizado, a gente vai aperfeiçoando esse template a cada vez que tem uma dissertação escrita em inglês, né? Então, o Rodrigo vai ser mais uma dessas pessoas que vai ajustar para ficar mais ou menos perto do padrão que, infelizmente, a nossa instituição não definiu, né? Então, a gente está fazendo ele no dia a dia, né? Então, alguns desses problemas na estética da apresentação tem a ver com essa indefinição local, né? Agora é só isso. Obrigado. Rodrigo, pode usar a palavra aí.
Certo. Eu não preparei um discurso, eu só queria agradecer todo mundo, né? Agradecer ao professor pela oportunidade. Também agradecer a Arthur, que me ajudou bastante durante esse período, né? De mestrado e até antes dele. Agradecer também ao pessoal do laboratório, o Anderson, e também agradecer a Matheus, que eu acho que ele saiu da apresentação, mas ele estava até agora há pouco, eles me ajudaram bastante também. E agradecer a todo mundo, né, que eu tive contato durante ele não está aparecendo, mas durante esses anos de mestrado, dizer que foi, acho que um momento bem conturbado, bem difícil, mas que graças a Deus eu consegui concluir, estou conseguindo concluir, né, com muita dificuldade, mas está dando certo, né. Acho que valeu a pena e acho que eu já estou pronto para as próximas, né. Acho que é só isso mesmo, professor. Sim, agradecer também a minha família, né? Agradecer meu pai, meus irmãos por todo o suporte e agradecer minha noiva também, que inclusive veio assistir aqui presencialmente. Ela está assistindo. E também sem ela eu acho que eu não conseguiria ter êxito, né, nesse trabalho. Agradecer meus primos também que estão assistindo, Felipe, Gustavo. E meus amigos também, que também estão comigo desde do colégio, né? Igor, Márcio, João Arthur, que também assistiram. Agradecer a todo mundo, né? Acho que é isso.
Ok, então, Rodrigo, parabéns mais uma vez. Boa continuação dos estudos, né? Você vai para o doutorado.
Sim.
E é isso, então. Alguém mais gostaria de fazer alguma colocação?
Eu quero falar um pouco, professor, se me permite.
Sim, professor, à vontade.
De novo, parabenizar, Rodrigo, aí pelo teu trabalho. Eu sei o quão extenso foi e o nível de complexidade dele. Parabéns, realmente. Espero que você continue aí na área. Tem muito o que gerar frutos ainda esse trabalho que você fez até então. Então, parabéns, viu? Parabéns realmente aí.
Obrigado, professor.
Pronto, senhores. Então, não havendo mais nenhuma manifestação por parte dos que estão presentes nessa sessão, podemos encerrar, né?
Exatamente.
Então, até a próxima, se Deus
Até a
quiser.
próxima.
Até a próxima a todos.
Até mais.
Até mais.